Hello friends, welcome back to my YouTube channel. Today in this video, we'll be going to talk about the gene therapy. What is gene therapy? Gene therapy is nothing but where the gene is transferred. Human gene therapy is nothing but where the gene is transferred to treat disease rather than drugs and surgery. Okay, so rather than drugs and surgery. So here gene therapy was first performed in 1990 in a 4-year girl who is a 4 years old girl, okay, whatever maybe, who is severely suffering from SCID, severe combined immunodeficiency disorder. So it was successfully done, and gene therapy is of two types. It is of two types. One is somatic and other is germline. What do you mean by somatic? The name itself is saying that somatic cells, okay, so where the cells can be transferred, okay, cells can be transferred. It does not produce any gamete, doesn't produce any gametes, okay. Whereas in germline therapy, where it produces the gametes, okay, it produces gametes, either sperms or eggs, okay, sperms or eggs. So it is of two types. And also gene therapy may be of two types. What are those? Those are classical and non-classical. Where classical is also called as gene augmentation therapy. Okay, classical therapy is also called as gene augmentation therapy, whereas non-classical is also called as gene inhibition therapy. What happens in the classical therapy? We'll be going to see that. What happens in the classical therapy? In classical therapy, where here we will be introducing the gene, okay? Here we will introduce the gene so that it will replace the gene which is suffering from the disorder, okay. Here we are treating the disease. Here we are treating the disease or disorder, okay. So what we are doing, the gene which is suffering with disease, okay, disease gene, this is suffering with disease, and we are introducing the other gene, new functional gene, into the body, okay, and it is replacing the gene of the disease. Replacement of disease gene, okay, replacement of disease gene. So this happens in the classical therapy. Whereas in non-classical therapy, what happens? In non-classical therapy, the faulty means the gene here, cancer like diseases can be done. Obviously the gene which is getting faulted in our body, it will fight with our own cells, right? So that will be replaced with the new gene. That is not replaced with the new gene, but the block gene we will add into the body, okay, to the faulty gene. When we'll add the block gene into it, what happens? It will block the faulty gene. It will block the faulty gene. Now if we talk about the gene target cells. So actually gene will target in two ways, okay. It will target the cell in two ways, that is ex vivo and in vivo. Okay, ex vivo means, ex vivo name itself is saying that we are culturing outside. Okay, so we will take out the tissues which can we take out from the body of human or any other thing. Will be take out the tissues from the body and we will culture it, and the clone one will be transferred to the patient. This happens in the ex vivo. Whereas in vivo, what happens is, here we cannot able to take out the tissue from the body, and we have to culture the gene, the introducing gene, we have to culture it in the body itself. Not culture, it should have to replicate in the body and cloned in the body itself. Okay, cloned in the body itself, that we call it as in vivo. So there are the two process by which we will introduce the gene in the body, that is ex vivo and in vivo. Hope you understand about this, and if you are new on my channel, please subscribe my channel, share my channel with your friends. Thanks for watching.